found in a cell is determined by mechanisms that turn on or turn off transcription of the genes that are coding for those proteins. In this way, cells are able to efficiently regulate the use of their energy and resources and only produce the proteins that they need for whatever function they're serving at a particular time. But in eukaryotic cells especially, with their complex internal organization, including specialized organelles and other compartments, it's not only necessary for the cell to produce the right set of proteins, it's also necessary for those proteins to get delivered to the right place. For example, proteins involved in replication and transcription of DNA, such as the enzymes DNA polymerase or RNA polymerase, and the many proteins that work with them, have to be located in the nucleus of the cell because that's where those molecular processes naturally occur. Similarly, other kinds of proteins might have to be located in other organelles, or perhaps even embedded in the cell's membrane. Still, other kinds of proteins have to be exported from the cell, secreted to the outside so that they can serve as, for example, molecular signals to other cells, digestive enzymes in the gut, and so forth. Thus, another issue that we have to address in order to understand how cells become specialized for a particular function is to ask how proteins find their way to their proper location inside or outside the cell after they've been synthesized. Now, you might think that one way this could happen would be for proteins to simply diffuse randomly throughout the cell after they're produced. After all, the cell's not really a very big place, and you might guess that eventually they'll show up in the right place and just stay where they belong. But there are two major difficulties with this idea. The first problem is that even in a space as small as the size of the cell, random diffusion just isn't a very efficient process. Imagine a store in which the stock boys wander around aimlessly, just placing items on the shelf wherever. Some items would end up in the right places, but most wouldn't, and it might take a very long period of time to completely stock the place where a particular item belonged. Now, the second problem is even more serious. Many of the places where proteins need to go on the inside of the cell are actually inside the various organelles of the cell, and thus inside compartments that are themselves sectioned off by biological membranes. Now, most molecules, especially large electrically charged molecules like proteins, simply can't cross cell membranes on their own. Instead, they need to be somehow transported across the membrane, for example, by another molecule that acts as a portal, or perhaps packaged in their own little membrane-bound container, a small, what we call a vesicle, which will take them across the membrane by fusing with the membrane of the organelle where they're being delivered. Now, to overcome these problems of getting delivered to the right place in a timely fashion, cells have developed a number of what we call protein trafficking mechanisms that are specifically responsible for moving proteins around in cells and getting them to the right place. Our goal in this lecture is to begin to understand how some of these mechanisms work. Early on in the course, you'll recall, I described the structure of eukaryotic cells to you when we were talking about the importance of cells as a unit of organization in the hierarchy of living things. I want to begin today's lecture by going back and spending a little time reminding you and expanding on what we talked about then. Remember that eukaryotic cells are characterized by having many internal compartments that we call organelles. The significance of this compartmentalization, you'll recall, is that it allows the cell to sequester different substances or different biochemical processes and keep them separate from the rest of the cell. In so doing, the cell is able to manage a far greater number of activities than it could do if everything was just all bunched together in a single common space. This is a little bit like a chemical company having different vats for manufacturing different products. It usually won't do to try to run several processes all in the same container. Now, one of the sets of organelles that I described to you earlier is called the endomembrane system, and this is what I want to review in a little more detail right now. If you look at a typical eukaryotic cell under a good microscope and with the right staining, the most obvious structure you see is the nucleus, of course. This is surrounded by the nuclear envelope, and this is where the DNA resides. Now, the next most obvious thing you'd see, just outside the nucleus and extending throughout the rest of the cell, is a series of internal membranes that spread through the cell, creating what look like a series of interconnected compartments. These compartments look like flattened tubes or pancake shapes. This set of compartments is what we call the endoplasmic reticulum. The inside of the endoplasmic reticulum is indeed a continuous set of chambers, like a big maze in the cell, and it's separated from the rest of the cell by its own membrane, similar to the membrane that surrounds the entire cell. Now, if you follow the endoplasmic reticulum further out towards the periphery of the cell, the chambers begin to look like they're breaking up a little bit. Now, in a real cell, if you looked at it in real time, the structure of the endoplasmic reticulum is dynamically changing all the time. But if you look at a snapshot, which is essentially what you're doing when you prepare a cell to look at it under the microscope, it might look like a number of small membrane enclosed bubbles have broken off from the ends of the endoplasmic reticulum's chambers. In fact, this impression isn't far from the truth. These bubbles, which we call vesicles, are budding off from the endoplasmic reticulum. By the way, a short way of saying endoplasmic reticulum is to simply call it the ER, and I might do that every once in a while. Now, even further out, beyond the endoplasmic reticulum, you see yet another stack of large pancake-like chambers. These are similar in some respects to the ER, but differ in the fact that, unlike the ER, each pancake is actually physically separated from the other. This second set of chambers is called the Golgi apparatus, which is named after a famous Italian microscopist, Camillo Golgi, who first described it back in the late 19th century. Many of the vesicles that bud off from the endoplasmic reticulum look like they go and then fuse with the chambers of the Golgi apparatus, suggesting that these vesicles might form a functional connection between the two structures. In fact, vesicles also appear to bud off from the other side of the Golgi apparatus itself, and these vesicles appear as though they travel to other organelles or to the outer cell membrane. The overall impression that you get when you look at the structure of the endomembrane system under a microscope is that the endoplasmic reticulum and the Golgi apparatus form an enclosed set of passageways, either directly interconnected, such as the passageways of the endoplasmic reticulum, or functionally connected by little transport shuttles, these vesicles, which appear to move from the ER to the Golgi and from the Golgi to other parts of the cell. Now, there's one more feature of the endoplasmic reticulum that I need to mention at this point. In some places, the endoplasmic reticulum looks kind of smooth. We call this smooth ER, not surprisingly. But in other places, especially closer in towards the nucleus, the endoplasmic reticulum looks very rough on the surface. Hence, we call it rough ER. An even closer look at rough ER reveals a very interesting thing. The rough ER is rough because its surface is covered with ribosomes. Now, remember from our earlier lectures in the first part of the course that ribosomes are the protein building machines of the cell. Ribosomes are found throughout the cytoplasm of the cell, but many ribosomes in the cell are found to be physically attached to the outside surfaces of the endoplasmic reticulum. Those parts of the endoplasmic reticulum we call rough ER. Now, the anatomy of all of this on a microscopic level, including the association of ribosomes with the ER and the existence of vesicles that appear to bud off from the ER and the Golgi apparatus, has long suggested that at least one function of the ER, of the endomembrane system in general, is to process, package, and transport proteins after they've been synthesized. This hypothesis was tested explicitly by experiments done in the 1950s by George Palade and his colleagues who were at the Rockefeller University. Palade knew that some kinds of proteins are manufactured by ribosomes that are just in the cytoplasm itself, and that these proteins remain in the cytoplasm. He also knew, however, that other kinds of proteins are secreted to the outside of the cell, especially proteins involved in digestion, for example. Palade hypothesized that these proteins, the ones that are secreted, are the ones that are synthesized by ribosomes attached to the endoplasmic reticulum. And he suggested that the growing polypeptide is essentially being fed into the inside of the ER as it's being synthesized. Palade further suggested that the newly synthesized polypeptide then moves through the ER, out through the Golgi apparatus, and eventually gets packaged into vesicles that transport those proteins elsewhere, such as to the outside of the cell. To test this idea, Palade's group used cells from the pancreas of guinea pigs. Now, they knew that these pancreas cells were specialized in secreting digestive enzymes. That's pretty much all these cells do. And thus, these cells could be expected to manufacture and transport large amounts of proteins that, according to Palade's hypotheses, should be transported through the endomembrane system. How do they test this hypothesis? Well, they developed a method for growing pancreatic cells in a cell culture, that is, in a dish without the rest of the animal. They would add to this cell culture radioactively labeled amino acids. These radioactively labeled amino acids would be taken up by the cells and incorporated into any proteins that were being synthesized. And thus, the proteins being synthesized would be labeled with radioactivity. Now, the trick here is simply to find out how to follow the fate of newly synthesized radioactively labeled proteins. Palade did this by adding the radioactively labeled amino acids to the cell culture for only a very, very brief period of time, a couple of minutes at most. And then after he had added the radio-labeled amino acids, he would wash them out of the cell culture and replace them with unlabeled amino acids. In this way, only proteins produced during a brief point in time became labeled. Now, the idea here is that you can essentially follow this very small pulse of labeled proteins as it moves through the cell, even though other proteins are continually being synthesized. Think of this like following packages on a complex set of conveyor belts that are moving through a large manufacturing plant. If all the packages look the same to you, and if there are a lot of places they could go on these conveyor belts, it would be very difficult to trace the path of any one package because you simply can't keep track of it. But if you could label one package a different color, paint it bright orange, for example, then you'd have no trouble in following that one package and in so doing trace where the conveyor belt goes. This is exactly the idea behind Palade's experiment, where just a small group of proteins were labeled, and then he could follow them through the cell. Following them through the cell by using microscopic techniques that allowed him to actually see where the radioactivity was. Now, if he looked for the radioactivity just a few minutes after he introduced the pulse of radio-labeled amino acids, he found that the radioactivity was all located just on the inside of the rough endoplasmic reticulum. If he waited a few minutes longer, the radioactivity had moved further out the endoplasmic reticulum, out towards the periphery, towards the Golgi apparatus. Maybe 20 minutes or so later, the radioactivity appeared in the Golgi apparatus itself, and if he waited an hour or so, the radioactivity would appear in vesicles that were budding off of the Golgi apparatus. All consistent with the hypothesis that indeed the endomembrane system is a system that transports proteins that are being manufactured. Now, Palade's work demonstrating how proteins move through the endomembrane system brings us to the next question we want to address, which is, how do proteins actually know where to go in the cell? Now, one obvious question is, how do particular proteins get directed into the ER when they're being synthesized while others aren't? Now, another thing that became clear from the work of Palade and many other cell biologists was that proteins that are produced and packaged in the ER may end up in very different places. The vesicles that bud off of the Golgi may end up in a number of places, different organelles, or even outside the cell. So this observation leads to a second question, which is, how do the vesicles that actually bud off of the endomembrane system actually get targeted to go to their appropriate destination? Let's start with the first question. How do particular proteins get directed into the ER when they're being synthesized? A novel hypothesis for answering this question was first proposed by Günter Blobel in the early 1970s. Blobel was a German cell biologist who had joined Palade's laboratory at the Rockefeller University several years earlier. Blobel suggested the following. He suggested that a sequence of amino acids occurring at the very beginning of a polypeptide serves as kind of an address tag, a tag that helps direct the ribosome that is beginning to synthesize that polypeptide to move to the endoplasmic reticulum if that's where the polypeptide should be synthesized. In other words, Blobel suggested that part of the amino acid sequence of the protein itself provides information about where that protein should go. Blobel called this idea the signal hypothesis. Blobel's signal hypothesis was actually consistent with an interesting observation that had been made by other researchers. As I said earlier, some types of cells, such as those of the pancreas, produce copious amounts of proteins that are exported from the cell. Now, the work of Pallotti and others had shown that these proteins move through the endomembrane system, but you can nonetheless cause these proteins to be manufactured in a cell-free system. You can take pancreatic cells, gently break them up, such as by agitating them in a blender, and then extract various components, notably the messenger RNA that's going to be the code for the proteins, and ribosomes, the protein builders. If you put these components along with amino acids in a test tube under the right conditions, without endoplasmic reticulum, you can still synthesize the proteins. But when that happens, in the absence of the endoplasmic reticulum, the proteins that are synthesized end up being about 15 or 20 or maybe 24 amino acids longer than they would be if they were synthesized in a normal cell. Well, how does that observation make us think differently about Blobel's hypothesis? In other words, how does the observation that proteins are longer when they're synthesized in the absence of endoplasmic reticulum inform us about the role of signaling and how signaling gets a protein into the endoplasmic reticulum? Well, Blobel suggested that the first 20 or so amino acids that were normally serving as an address label would be removed by the endoplasmic reticulum if that protein got synthesized with the endoplasmic reticulum. These initial amino acids are removed because their only function, Blobel argued, is to serve just as an address tag. And once that addressing function is done, the protein being synthesized in the ER has to remove those amino acids because they're not a part of the functional protein. This idea could be tested now by going and adding endoplasmic reticulum to our cell-free system. So we're adding the messenger RNA, we're adding ribosomes, we're adding amino acids, and now we're also going to add some intact endoplasmic reticulum in our test tube. And when we do that, we find that the proteins that are synthesized now indeed are the same length as the proteins normally synthesized by the cell. In other words, those first 20 or so amino acids have been removed by the endoplasmic reticulum itself. Quite a few experiments over the last several decades have gone on to provide very strong support for Blobel's signal hypothesis, and also to show how this amino acid address tag works. Let me just summarize some of this work. First of all, we now know that all protein synthesis begins on freestanding ribosomes in the cytoplasm. However, if the protein begins with a specific address tag, then that ribosome, once it's finished synthesizing those first 20 or so amino acids, stops synthesis. Those amino acids will cease the synthesis of that particular polypeptide. Those first 20 amino acids then serve to attract another molecule called a signal recognition particle that attaches to those amino acids. And the combination of the addressing amino acid and the signal recognition particle then directs the ribosome to the endoplasmic reticulum and docks it on the endoplasmic reticulum. Once the ribosome has been docked on the endoplasmic reticulum, synthesis begins again, but now the protein being synthesized is directed to the inside of the endoplasmic reticulum. And as that happens, that first sequence of amino acids is clipped off and thrown away. The important take-home message here is as follows. The address label that determines whether a protein goes into the ER or not when it's being synthesized is part of the protein itself, a part that is thrown away when its job as an address tag is completed. This means that the information in the label, which ultimately determines where that protein goes in or out of the ER, is actually coded for by the DNA for the gene for that protein itself. The code for the first 20 or so amino acids is a code for an address tag that's different, or at least not necessarily a part of the code for the functional part of the protein, because these first 20 amino acids are discarded before the protein is completely manufactured. Well, labeling proteins as to whether they should go in or out of the endoplasmic reticulum is only one part of the problem we have to solve. Remember, we also have to ask how proteins get put into all of their other final destinations. Let's look at an example of how this might happen by considering what happens at the other end of the endomembrane system. Remember that the proteins synthesized in the ER move through the endomembrane system and are eventually packaged in vesicles by the Golgi apparatus and delivered to their final destination there. How is it that those proteins packaged in vesicles end up in the right place? Well, as proteins are moving through the endomembrane system, a number of things happen to them. They're processed by a whole series of enzymes inside the endomembrane system that essentially polish them off, change them in a number of ways. They might remove some amino acids, as we saw with that signal sequence that was clipped off. They might change their shape in certain ways, helping them to fold up, or they might actually modify some of the amino acids chemically that are part of the protein. One common thing that happens to many kinds of proteins as they're processed in the endomembrane system is to have various kinds of carbohydrates, those are sugars, added to them. This process is called glycosylation, which basically just means sugar addition, and the resulting protein is in general called a glycoprotein. This sugar coating of proteins can have a number of different functional consequences, such as helping the protein to fold into its right shape or stabilizing it if it needs to be stored. Additionally, the sugars that are added to the amino acids of some proteins often act as another kind of address label, a label that determines where that protein will go after it leaves the Golgi apparatus. Let's see how this worked by considering the fate of proteins destined to go to an organelle in the cell called the lysosome. Now, if you look at the anatomy of the cell again, lysosomes are actually fairly boring-looking things. They're just large vesicles, large round vesicles without a lot of structure. Lysosomes are sort of like the stomach of the cell. Lysosomes are filled with very powerful digestive enzymes. When a cell takes in a very large molecule that it's going to use as food, these molecules need to often be chopped up before they can be processed by other parts of the cell. Just the same way that food is essentially chopped up in your stomach before it's transported onward into your intestine for further processing. Now, obviously, the enzymes that do this chopping up have to be sequestered in their own space, like the lysosome. Otherwise, they'd go and they'd chop up all of the other proteins and components of the cell itself. The cell would digest itself. The digestive enzymes that end up in the lysosome do come from the endomembrane system, and they're packaged in vesicles that are specifically targeted to go to lysosomes. But here's the problem. There are all sorts of proteins in the Golgi apparatus that are going to different destinations, and only some of those proteins, these enzymes, are destined to go to the lysosome. How do the right proteins get put into the right packages and have those right packages go to the right destination? Well, the answer has to do really with a pair of signaling molecules. The proteins themselves have sugars added to them which serve as their own kind of address label. These sugars will bind to membrane-bound proteins on the surface of the Golgi apparatus on the inside in a very specific way. Particular kinds of sugars will only bind with particular kinds of membrane-bound proteins. These membrane-bound proteins themselves, in turn, will serve as a kind of address label for the vesicle when it eventually buds off from the Golgi apparatus. So how do the right proteins get put into the right vesicles? The first step is those proteins have sugars added to them which address them to a region of the Golgi apparatus's membrane that will turn into a vesicle going to a particular target. Then those membrane-bound proteins, which originally attracted and bound the sugars of the proteins being packaged, the proteins on the surface of the vesicle themselves, will in turn serve as an address label for the vesicle. So these membrane-bound proteins have kind of a dual addressing function. On the inside, they attract the right sugar address label to get it packaged into a particular vesicle. And on the outside of the vesicle, after they've budded off, they serve as the address label for the vesicle itself. Well, there are a lot of details about how addressing works in proteins, getting them to the various parts of the cell, but these examples give you the general idea. Proteins have labels. The packages they're shipped in have labels. The labels themselves might be part of the protein, or they might be proteins whose only function is to serve as an address label, such as those address labels for the packages that are transported to the lysosomes versus other parts of the cell. The proper addressing of proteins, to make sure they get to the right place in a cell, is as important for normal cell function as the ability to produce these proteins in the first place. I want to end today's lecture by briefly giving you an example of what can go wrong if proteins aren't correctly targeted to their destination, and this in the context of the proper functioning of lysosomes. Inclusion cell disease, or sometimes called eye cell disease, is a genetically inherited condition that is very debilitating. It's relatively rare, but individuals who suffer from it have severe skeletal abnormalities when they're developing as children, as well as mental retardation, and it usually results in death before the age of 10. Interestingly, inclusion cell disease is caused by malfunctioning lysosomes. What happens is that individuals with eye cell disease are missing critical digestive enzymes from their lysosomes. Food particles get sent to the lysosomes, but there are no enzymes there to digest those food particles. The lysosome gets filled up with a bunch of stuff, grows, becomes very large. These are the inclusions that you see in the cell itself, which is why it's called inclusion cell disease. And ultimately, these packed up lysosomes, these malfunctioning lysosomes, will destroy the cell's function. Inclusion cell disease is genetic. It's known to be caused by a single recessive mutation. So we then can conclude that inclusion cell disease is probably caused by some malfunctioning protein. Well, what are the possibilities? Now, based on what we know about molecular genetics and mutation, the first thought we might have is that inclusion cell disease is caused by a mutation in the digestive enzyme itself. Perhaps individuals with eye cell disease just simply can't manufacture the right digestive enzymes, and that's why those digestive enzymes aren't found in the lysosomes. Another possibility, based on what we've learned, is that inclusion cell disease is caused by a mutation not in the protein, the enzyme protein itself, but perhaps in a protein that is regulatory for that enzyme, a transcription factor that controls the production of the digestive enzyme. Here again, a mutation in the transcription factor might mean that the individual can't produce the digestive enzyme. But neither of these possibilities prove to be the case. Instead, if you look at patients with eye cell disease, if you look at their cell function, you find that they are capable of manufacturing these digestive enzymes. It's just that those digestive enzymes never get out of the Golgi apparatus. So they can make the enzymes, they can't get it to the vesicles. Well, why not? Well, the enzymes that are found in individuals with eye cell disease are different from those found in normal individuals in one particular way. They lack a sugar that should have been added to them, a sugar called mannose 6-phosphate, not an important detail. But it turns out that this sugar, mannose 6-phosphate, is an address label. That sugar, when it was attached, if it is attached to the protein, is the address label that will make that protein find the right vesicle, the vesicle that will transfer it to the lysosome. If that address label is missing from the protein, the enzyme, can never find its way into the right package, and as a result, it never gets sent to the lysosome. The lysosomes don't function properly, and serious consequences ensue. Well, now that we've looked in detail at how cells control the kinds of proteins they make and how they get them to where they belong, we'll next take a look at how cells communicate with each other, another prerequisite for the ability of cells to develop into multicellular organisms and the ability for cells within an organism to coordinate their activities. But before we do that, we're going to take a short detour in the next lecture to look at how our knowledge of molecular genetics can be put to some practical use through the use of genetic engineering and biotechnology.